I'm Elizabeth Regan from the College of Engineering and Computing at the University of South Carolina. Welcome to our session on using artificial intelligence and knowledge graphs for precision medicine at scale. It is my pleasure to introduce our speaker for today's presentation, Dr. Sergio Baranzini. Dr. Baranzini is a distinguished professor of neurology at the University of California, San Francisco. He's also a member of the graduate program in bioinformatics, the Institute for Human Genetics, the Becker Computational Health Sciences Institute, ImmunoX, and the California Institute for Quantitative Biology. He holds the Heydrich Friends and Family Endowed Chair in Neurology. Dr. Baranzini earned his degree in clinical biochemistry and a Ph.D. in human molecular genetics from the University of Buenos Aires, Argentina. Dr. Baranzini then moved to the University of California in San Francisco to specialize in the analysis of complex hereditary diseases and focused his efforts on multiple sclerosis. There's lots more to his bio, and I encourage you to take a look at his profile if you'd like to learn more about his background. But now I'd like to turn the floor over and with a big welcome to Dr. Sergio Baranzini.
Well, good morning, and thank you very much for the kind introduction and for the invitation to speak at this conference that, from what I've been monitoring, it is very active and it looks great. I was so looking forward for this to be in person, but this is the next best thing, and I'm really excited to be speaking to you all today. So let me start sharing my screen and we'll get right down to it. I need confirmation that you're seeing my full screen.
Yes, we see it.
Perfect. Well, so today I'm gonna be sharing some of the philosophy, then research and some unpublished results that we've been taking into this relatively new topic, which is the use of AI and in particular knowledge graphs for precision medicine. And let me start by saying that we really follow the tenet that the data leads to information, and information leads to knowledge. And I'll like to stop here and define data and information particularly, because usually they're utilized in interchangeable ways, whereas I like to make a difference, because it really matters when we're putting together different kinds of information to generate perhaps knowledge. So data, when we refer to data, is individual experiments. Data are facts, that are figures, are not necessarily organized or synthesized in any particular way. Information is when that data becomes aggregated and now you have more evidence for a particular process, for a particular interaction, for a particular assertion to be made. So information is somehow synthesized data. It requires more than a single experiment to be able to make that assertion. And information may become knowledge when that refers to connecting those dots, to connecting the dots of information, when it refers to know-how, it refers to understanding. In a sense, knowledge is an emergent property of a large web of information, and that's why we equate this with knowledge graphs, which is the topic of my presentation. Of course, you can continue this in some cases. People have started thinking, well, maybe you can also define insight in this way, when it's exactly, you know, the path from one place to another place in your cognitive network. You can say, well, wisdom is when you don't take any other path than that path, and then you have achieved wisdom. Some scholars have gone too far and have proposed additional uses for this schema, but we'll not get into that right now. So the topic of my presentation involves biomedicine, and in biomedicine, I think most of us will agree that if we create a sort of a hierarchical organization of biological complexity, we could start from the bottom up with genomic information or metagenomic information right now. Then the next level up will be that of gene expression. Transcripts will get translated into proteins. Proteins will interact, and they will form pathways, biological pathways that will be interpreted by cells. Cells will integrate that information, all that stimuli, and then they will produce an output. And the output of that cells might be differentiation or apoptosis, cell death. And then cells organize into tissues, into organs, and then all the way up we have the organism, which is what a doctor will see in his office or her office when examining a patient. But rarely will anyone be able to navigate with the same level of confidence across different strata as depicted in this illustration. And most likely people will spend their entire, if you're doing research, you probably will spend the entire career looking at just one of this. So if you're a biochemist, you're going to spend most of your time here. If you're a cell biologist, you're going to spend most of your time here, and so on. So, but now I think there's enough information accumulated in the different strata that is probably good time to start integrating and to start producing tools that will allow an investigator or a medical professional to navigate across well-established areas of knowledge up and down this hierarchy with the confidence that, yes, this is textbook, almost textbook information, but it's all linked. It's not just an internal medicine textbook or a biochemistry textbook. It is a textbook of everything we know. And that will enable a large number of applications, including some of the ones that I'm going to talk about. So when I talk about complexity, I mean this kind of complexity. And this is just one of those levels. This is just a metabolic map of humans. And as you can see, it's an incredible amount of feedback loops and connections. And how do you make this computer readable so that they can be computed over and then human interpretable? So that's the challenge that we have in front of us. And this is why we built Spock. Spock is a knowledge graph that represents information accumulated or stored in more than 40 different specialized databases. Spock contains more than 20 million nodes and more than 50 million edges or connections among those nodes. And Spock has been really started in my lab more than 10 years ago. More recently has been funded by the NSF, and we really gave it a boost with all our partners from ISB, Lawrence Livermore National Lab, UC San Diego, Stanford University, and Indiana University. And now so this is the view that you're seeing here is what we call a metagraph. So this is just a 30,000 foot view of the kinds of nodes and relationships that the graph contains. And so where it says gene here, that means every gene in the human genome is represented. And when we say there's a link between genes and diseases, well, that means that every single genetic association that we know of and that is stored in reputable databases will be represented in this graph. And when we say disease, there's more than 10,000 diseases stored in Spock. When we go to symptoms, then we describe the symptoms of those diseases as described in databases or textbooks. Then there's relationships to pharmacological compounds. There's relationships to food. There's relationships to the side effects of those compounds, the pharmacological class of those compounds, and so on. We have an entire section on metabolic pathways. And if we're talking about human metabolic pathways, we cannot ignore the microbiome. So we also introduce more than 5,000 microorganisms that are of human importance. Although Spock is human-centric, again, we are about 50% bacteria by cell count, so we cannot ignore the contribution of the microbiome. So, and I'm going to show you some examples of this and how you can navigate it and how you can make discoveries. So this is just again a bird's eye view of all the diseases, and you can do this through our graphical user interface that I'll show you later. You can search by any disease and then get the neighborhood around that disease, and if you so choose, you can get only diseases that are neighbors of that. And diseases in this case are linked to other diseases by virtue of the disease ontology. So we use ontologies, which are control vocabularies and are built in a way that they form a directed acyclic graph, so that there's no feedback loops back. So a parent category has several children category, and this is a perfect description for hierarchical, for information that is hierarchical in nature, such as diseases. So you get the top node will be disease, and then the first level below that might be neurological disease or inflammatory disease or muscular disease, and then below that you start getting more and more details. So you can navigate the entire ontology for diseases. We have ontologies, of course, for genes, ontologies for pathways, ontologies for food, because ontologies help us anchor the different kinds of informations that we retrieve from the different databases and help us map the concepts in a semantic, meaningful way. The next example that I want to show you is the bacterial taxonomy. So here what you're seeing is more than 5,000 bacterial species, and here the taxonomic level is color-coded, so you can go all the way up from phylum on all the way down to species. And then here is the metabolic pathway representation. So again, you can navigate through any of these metabolic pathways. In this case, you say, okay, there's a carbohydrate metabolism here. Within this category, we have the central carbohydrate metabolism. Within this category, we have several pathways that may be represented by different organisms, particular humans or different bacterial organisms. And then one of them might be glycolysis. And then if you click on this, you get the entire glycolytic pathway. So pathways are modeled in a way such that the metabolite is described here in blue, and then the reaction that either consumes or produces a metabolite is depicted in pink. So you can navigate from one compound through a reaction to the product, and then this product in turn may become the substrate of the next reaction, and it will be and this will be the product of the second reaction, and so on. So you can navigate entire pathways in this way. So I mentioned about a graphical user interface. We call this the Spock Neighborhood Explorer. Here's the URL below, and Spock can be accessed both by this URL and also by an API. So the kinds of things you can do in Spock are exploring the neighborhoods of particular search nodes. And here you have a text search that you can first decide what you want to search for. You can search for a compound, a disease, a gene, a pathway, an organism, a food, and so on. And then you type in your search in the box, and then you click submit. And then based on the options that you can decide on, you're going to get a neighborhood, and you can choose to retrieve everything that Spock knows about your particular query term, or you may want to limit the options, which is what I would recommend at the beginning. Limit the options to certain types of nodes or edges that you want to see. Otherwise, it may be overwhelming, and they may become, remember, there's 20 million nodes, so you don't want to bring too many in a particular visualization. But let me show you an example of what this can do. In this case, these are all the proteins encoded by SARS-CoV-2, the virus that causes COVID-19. And so each of the viral proteins is depicted as a light blue circle, and then all the interactions, all the interactions to human proteins is described as links to these green nodes. So if we zoom in one of them, here we have the spike protein of the virus, and these are the 3 human proteins that have been shown to interact physically through experimentation, pull down and mass spec approaches and immunoprecipitation to describe the interactions between the viral protein and the human protein. And of course, ACE2 is the human entry point of the virus. So we can describe this, and then we can go one step forward further and say, okay, so in addition to the protein interactions, I want to know the genes that encode those proteins. So for each of these proteins, there's a gene that encodes it, and we make a distinction between genes and proteins because some relationships are much better explained in protein space, for example, protein-protein interactions, and some relationships are better explained or represented when the information comes out of the gene, for example, gene regulation or genetic predisposition to a disease. So we can go one step further and say, okay, now I want to bring compounds that are known to bind to ACE2 human. And here you have captopril and enalapril. These are drugs that are normally used in cardiovascular diseases because, again, ACE2 is a critical component of the renin-angiotensin system, and that helps control blood pressure. And these are medications that are known to have an effect on these conditions. So we can go one step further and say, okay, what are the diseases now that are linked specifically to this gene ACE2? And here you have a number of them. I don't know if you can read them, but you have several cancer diseases and several urinary system diseases and so on. And of course, cardiovascular disease. Then we can say, okay, I want to know where this gene is normally found within the cell. So we're bringing the cellular components of those of that gene in the yellow nodes. For example, this gene is expressed at the cellular surface. And then we can bring in molecular functions. So what is the described molecular function of this gene? We can also add, where is this gene expressed? So which are the cell types or the anatomical sites in green where this gene is expressed? So you can see, you see how you can start decorating your original search term with additional information. And you always know exactly where that information comes from. If you hover the mouse over, this is not live, but if this were live, you will see that if you hover the mouse on top of any node or edge, it will tell you what type of node that is, where it comes from, and some additional information such as a p-value that describes in more detail that particular association. So let me show you some concrete examples of what we have been using Spock for in certain areas. So of course, COVID-19 was one area that we turned our attention to. And about a year and a half ago, this paper made a big splash because this was Oak Ridge National Lab's supercomputer, where scientists in Oak Ridge put together a plausible model of mechanistic pathogenesis of COVID-19 and suggested possible therapeutic interventions. The way this graph was constructed was not from the bottom up, as I described Spock, but the way that this was constructed was recovering all possible information from the literature, mostly, and doing really text mining at petascale. And what they came up with was this representation of, here you have some genes, you have proteins, you have metabolic pathways, you have cellular signaling pathways, and the most of the processes that were highlighted here have to do, again, with regulation of blood pressure, with the renin-angiotensin system, with fluid intake, and so on. And one of the particular take-home messages of this paper was the discovery that perhaps part of the mechanism by which COVID-19 produces that difficulty in respiratory output is that it involves a pathway known as the hyaluronic acid, and hyaluronic acid is a substance that takes up fluids up to 1,000 times its volume. So it's really hydrophilic, and it may lay down on the interstitium of the lungs, and that may prevent the air exchange or the gas exchange between oxygen and CO2, and that may lead to the difficulty in respiration. So what we try to do with Spock is how well can we replicate this mechanistic model that was published, but using only information that we have in Spock, which we know it comes not from the literature, but it comes from actual experiments. And so we were able to replicate pretty much the whole pathway. So here is the SARS-CoV, the protein, and the activity of the receptor, all the way to involving the renin-angiotensin system, the coagulation system, the endocrine system, vasoconstriction, vasodilation, and all the way up to immune regulation, the cytokine storm, and the involvement of hyaluronic acid. So with this in mind, we were also able to start generating hypotheses. And one of the hypotheses that we generated using this was the fact that, and we published this in, you can see in May of 2020, the hypothesis was that in mechanically ventilated patients, we predicted that these patients will benefit from the use of dexamethasone, a corticosteroid. And the reason was that during mechanical ventilation, the stretching of the lung epithelium occurs, and the physical stretching of the epithelium triggers the release of a cytokine called midkine. And midkine is a cytokine that in turn unleashes a cytokine, and more important, a bradykinin storm. So what this does, it initiates this feed-forward loop in which more cytokines, more inflammation happens, and we knew from Spock that midkine was highly sensitive to dexamethasone. So we hypothesized that in these patients, the use of dexamethasone will be beneficial. Of course, this was a hypothesis. 2 months later, there was this paper coming out in the New England Journal of Medicine, in which a clinical trial had been performed in a hospital in London by the Recovery Collaborative Group, and they showed that indeed that was the case. Shortly after, both the FDA and the EMA approved the use of dexamethasone for patients under mechanical ventilation. So this is one of the examples. Let me now jump into another example in which now we're taking patient information into the mix. So far, everything that I've told you does not contain any patient information. It's just facts and experimental results, but there's no patient information in Spock. So this is through a collaboration with the UCSF Information Commons and the Precision Medicine Initiative. Both Atul Bude and Keith Yamamoto are great collaborators in this project. And through Information Commons, what we've been able to explore is the entire corpus of electronic health records from the UCSF patient population in a completely de-identified manner. So Information Commons has has stripped all the identifiers from everything, including the notes. We're not using the notes right now, but including the notes have been completely de-identified. And so now they're available for research. And what we did is we used almost 2.3 million electronic health records to embed them, and I'll show you how, into Spock and create an augmented electronic health record of the patient in the context of Spock. And this was work by Charlotte Nelson in the lab, a grad student in the bioinformatics program at that time. And what Charlotte came up with was an algorithm in which she mapped variables from the EHR on the left-hand side of a given patient. So a patient may have a genotype, or a patient may have a battery of lab tests throughout his or her encounters with the UCSF health system. A patient may have a diagnosis, of course. The list of medications will be listed, and so on. So throughout the history of encounters of a particular patient, there's dozens, sometimes hundreds of variables that can be extracted and that can be mapped one-to-one to nodes in Spock, because Spock contains every disease, every medication, every lab test, every metabolite. So then with this, Charlotte used an algorithm similar to the Google PageRank algorithm to diffuse the information from the patient across the network so as to create a unique vector that represents the status of that patient and gives at that point in time, and gives a quantitative assessment of the importance of each of the nodes in the network based on the input data. And she did this for every single patient in the UCSF electronic health record. So one of the things that we can look at now is we can visualize these vectors in a graphical output. And for example, this is a cohort of more than 4,000 patients with a post-traumatic stress disorder. And you can see the individual signatures of these embeddings for these patients. When you aggregate all of them, you get the signature of the disease. So the nice thing about this approach is that it's completely interpretable. You can use it in a machine directed way to learn about similarities and differences with other diseases. Indeed, this is what we did. We compare what would be other conditions that are similar to patients with PTSD, and these are the top 10. Bipolar disorder, nicotine dependence, schizophrenia, depression. So, and then we also compare to what would be the least similar condition to PTSD, and that was atopic dermatitis. So this makes sense. You can see the almost polar opposites in their pseudo-colored map. But like I said before, this is completely interpretable because we know exactly what are the underlying nodes in Spock that receive more traffic of information from embedding hundreds or even thousands of patients after patient into this. So now we can say, okay, which are the underlying nodes on the top 10 pixels of this image that will describe patients with PTSD? So if we zoom in, we can see nicotine at the center. We can see nicotine dependence as a condition. We can see all of the genes that have been genetically associated with nicotine dependence. We can see some of the side effects of nicotine and so on. So you get interpretable results by navigating and looking under the hood. So of course we can do this with all diseases. With here you can see 137 diseases, the individual maps of all of them, and you can start seeing patterns. So these in red are all the different cancers. You can see that there's similarities in the profiles, but there are also differences. So these are opportunities to dig deeper into each of those. Here are in green all immune-related or autoimmune diseases. Again, you can see similarities. You can see bipolar disorder here, autistic disorder, Alzheimer's disease, all neuropsychiatric diseases are in light blue, and so on. So this presents with an opportunity to do this type of analysis, which is interpretable, and then, as I will show you in the last section of the talk, there's predictive power to it. So in one of the most recent projects, what Charlotte did was created timelines of a subset of the patients in our database, all those patients that had a diagnosis of multiple sclerosis. So, and here you see the timeline of patients. This is when they received the first ICD-9 or ICD-10 code for MS. And then she created a timeline that goes all the way back to 7 years before that was. So then she created snapshots of patients at 1 year before diagnosis, at 3 years before diagnosis, and 5 years and 7 years. And of course, we get more attrition as we go back. And then she created a control population of patients who were never given an MS diagnosis code. And she did the same. And then she compared the two. She embedded all of this into Spock, and then she compared what are the probabilities that we can identify these patients before MS appears in the record. So at -1 year, how well can we distinguish this population from this population just from the Spock embedding vectors? And this is the results. This is at year -3. So this is 3 years before. And this was done using random forest classifier. And what we achieved was 83% accuracy in predicting who will be developing MS. And this is very important from the clinical point side as well, because as you may know, MS is a chronic disease. There are treatments for multiple sclerosis. However, the sooner you start treatment, the best outcome generally. So it's important to diagnose patients early and accurately. And this comes from primary care visits. So these patients haven't even consulted with a neurologist yet. So I'm not suggesting that this is a diagnosis method, but this is a screening method that if a primary care physician is confronted with this information, perhaps it will be appropriate to refer to the neurologist, and neurologist may want to have a brain MRI done. So another example is in Parkinson's disease. We did a similar, we took a similar approach, again, creating the timelines, trying to see if we can diagnose or identify the population that will be diagnosed with Parkinson's any length of time before the diagnosis actually happens. And this is an interesting step that we also did for MS, but I didn't show it. Before attempting prediction, what we could see is we can pick certain nodes in Spock and assess what is the importance of that particular node at the different snapshots. And for example, what we could see here is that even at 5 years before diagnosis is made, already the concept of Parkinson's disease, so the node representing Parkinson's disease in Spock, is already more visited, more important than all other nodes. And this is here shown as a -log10 of a p-value of a Wilcoxon test. So you can see that there's already some key nodes, cognitive disorder nodes, gait apraxia, which is disturbances in the gait. Tremor starts to show up significantly even 5 years before, and then it increases as the time to diagnosis approaches. In contrast, other concepts that are not related in any way to Parkinson's, such as the content of hemoglobin in urine or bronchitis or earache, these concepts are not significantly different between the population of patients that will be developing Parkinson's and the controls. So again, when we assess the performance of the random forest classifier for Parkinson's patients, we see that at minus 5 years, we have a modest but encouraging performance of 0.72. And again, as time to diagnosis approaches, this performance of the classifier increases. One of the things that we did also was to use a gold standard validation. And the gold standard validation is what happens if we give a clinician, an expert in Parkinson's, exactly the same information 5 years before or 3 years before that we're using to feed Spock. How well would a clinician be able to tell that that patient is Parkinson or is a control? And this is the performance of those classifications. In every single case, both at 5 years, 3 years, and 1 year, Spock predictions outperform the predictions of the neurologists, who did better than chance, but still worse than Spock. So in the last 2 minutes, I really wanted to tell you about a fun application, and this has to do with space biology. And we have a collaboration with NASA in which NASA collects a lot of biological information from their experiments that are done in the International Space Station. And in this case, what they did is they flown mice into the ISS for 30 days, and then they collected their gene expression information from different tissues, and then wanted to know what will be the pathways that were activated after 30 days in space. And of course, they could do the gene expression analysis. They have a wonderful system called Gene Lab to do this, but they came to us to say, can you link in any way these gene expression profiles to any higher order phenotype that we cannot tell from the mice, such as symptoms or side effects? And certainly, because we do have those kinds of nodes in Spock, we could do that analysis. And of course, when we look at pathways, we found similar results to what they had found doing regular pathway analysis of the gene expression profiling. And that has to do with DNA damage response, with the expression of defensins, innate immunity pathways, toll-like receptor, of course, cancer-related pathways due to the higher radiation and low gravity environment. But then when we look at symptoms, these are the symptoms, the top symptoms that came up when we analyzed the gene expression data. And we found edema, fever, abdominal pain, eye manifestations. We look at side effects. So we found nausea, vomiting, headache. So a mouse will never be able to tell you about this, but this makes sense because humans have reported this. So astronauts usually report these types of symptoms and side effects after space flight. So this was highly relevant, and we're continuing our collaborations with NASA to analyze other data sets now. So with this, I would like just to give you a sense of where we're going. Spock can, of course, be applied in many different situations. What I showed you today was the precision medicine approach. Of course, there's many more. We just spun off a company called Made Bioservices to make commercial applications of this. Spock remains open for academical use. And of course, we're interested in developing a much more user-friendly web interface than the prototype that I showed you. And we have a number of other projects, including drug repurposing and more precision medicine approaches. With this, I would like to acknowledge everyone who participates in this project, from our group, from different groups at UCSF, our collaborators externally, and our funding agencies, to which I'm really grateful. And I really appreciate your attention today, and I'm happy to take any questions. Thank you so much. I think you're muted.
Thank you so much, Sergio, for a wonderful presentation. Really exciting stuff that you're doing in terms of dealing with complexity. I mean, that's a growing research concern, right? In terms of the application of these technologies on the front lines, and the complexity when it translating research into practice on the front lines. So, you know, the whole approach you're taking in terms of, you know, the mapping is really exciting.
Indeed.
Exciting stuff.
Thank you very much. Indeed.
So there's one question here that a couple questions here that I'd like out. Can you see? I think I'm breaking up just a little bit. I'm having a little bit of trouble. Okay. This comes from Dr. Holatosi. Great presentation. How can researchers get access to Spock data to combine with existing claims data?
So Spock, again, can be accessed in two ways right now. One way is through the graphical user interface, and also this runs a REST API at the background. So programmatically can also be accessed through this API. So if you go to that page, there will be more information in there.
Okay. Can we display that again? Did you have that on the slide?
I'll copy the URL in the chat. Will people see it?
I can make sure they see it.
Okay.
In the chat, that's preferable. Okay. Yes. Okay. Good. All right. Okay. Miranda said she could do that for us. Okay. All right. Next question from Dr. Young. Thank you so much for this important work, Dr. Baranzini. Are Spock data, particularly for the patient EHR data, only available to those visiting the clinics, hospitals? Do you plan to capture patients' behavioral patterns from other sources, remote digital health data?
Yes. So at the moment, we have only processed data from the UCSF community. We just now got access to additional medical centers, EHRs, also in a de-identified fashion. We now have access to all of the University of California medical centers, which is more than 15 million records. We're very interested in collaborations with other medical institutions who may have access to this in completely de-identified fashion, so that we can validate across medical systems, because we know that it's important that we can show generalizability. So we're really interested in collaborations to be able to do this in other institutions and with other systems.
Another question here along that line from Anja. Have you heard of the Cray graph? Whoops, it just jumped on my screen here, sorry. Have you heard of the Cray graph engine? I recently read a paper where it seems like it is similar to Spock. If you do know of the Cray graph engine, how does it differ from Spock?
Unfortunately, I haven't read that paper, but I do know there is a number of similar efforts, and again, this is, I think, natural that the scientific community wants to do. It's a natural direction to go to. It was surprising to me that up until 5 years ago there was nothing in this area. However, there's many different ways in which knowledge graphs can be built. Most notably, there are approaches, and Google even has a version of this through its Data Commons platform. what they are attempting to do is, for example, using triples. In our case, we're using property graphs. There are some efforts that will take literature into account, some efforts that will decide to model the information in one way or another. I think right now the space is widely exploratory. There's no standard. It's mostly in the research realm, and I'm sure that there's a lot of different attempts. One key factor here that I didn't mention is keeping the graph up to date is challenging. As you know, at the moment you download data from a database, it becomes old. So in our case, what we're doing is we're writing code so that we download the database every week, and then the graph gets reassembled every week with the newest data, and then we expose the new data. And Spock continues to grow, so we're not done.
Okay. I may have frozen. Can you hear me? I think I froze. Okay.
Yeah, I can read some of the questions.
Yeah, we'll answer one more question. It's from Carolyn Williams, and she says, so many clinical databases and research databases exist that have in-depth data that are not in basic clinical data. How to sit data like that next to this would be very interesting to me. For instance, in-depth immunology data in HIV patient cohorts. Is this feasible?
Yeah, absolutely. So the examples that I've shown include only data coming from electronic health records, but as we all know, electronic health records, first of all, they were not designed for research, and they capture some information, but not everything perhaps that we would like to know. But there's no reason why we should limit our patient population to that present in the EHRs. We have a number of research cohorts that we're performing the same approach on. We have a 500 patient longitudinal cohort with multiple sclerosis patients, for example. We have access to a neuropsychiatric disease database. So any database that contains patient information of sufficient size, I think that will be the critical requirement there, and that contains information in identifiers that we can map. So the reason why we chose electronic health records is, and particularly in the EPIC system, is that they are mapped to the OMOP standard, and then from there we can enter Spock. So if there's a way that we can map any research cohort to the OMOP standard and then entering Spock, we can repeat these with literally any cohort. It could be just an Excel spreadsheet full of patient variables, and technically this could be doable.
Great, thank you. I think Betty might still be having issues connecting. So I will just take this.
All right, so okay.
Go ahead, Betty.
Okay, can you hear me? Okay.
Yes, we can.
Sorry, I apologize for that hiccup in my traffic on the internet, I guess. Do we have time for any more questions, or do we need to end?
We are out of time,
Okay.
but I
All
see other questions are coming
Yes.
in the Q&A on Whova.
Yes.
So Dr. Berensini can continue to answer them there. And I have put the link he shared in the chat box for everybody. Thank you, Dr. Berensini, so much for taking the time to join us today. It was a wonderful presentation. And please, everybody, join us again for our next keynote at 12:05. Thank you so much.
Thank you all.
Thank you, Sergio.